This lecture is going to cover the physiology of the parasympathetic nervous system. Previous lectures covered the anatomy of this system as well as the anatomy and physiology of the sympathetic nervous system. The parasympathetic nervous system is part of the rest and digest, so we're going to be looking at that and what kind of effects it would get from it. So let's just look at the arrangement of the pre- and postganglionic neurons, which was covered in the anatomy section, but we'll also add some neurotransmitters. So this cell body of preganglionic neurons, which you see is right here, are found within the central nervous system. So this could be within nuclei of the brainstem, and these are going to be associated with various cranial nerves, or it could be within the sacral spinal cord, but specifically S2 to S4. So the preganglionic neurons, which are fairly long, they come here and they're going to release the neurotransmitter acetylcholine. And on this, we have nicotinic receptors. So this is very similar to when we looked at the sympathetic nervous system, is all preganglionic neurons release acetylcholine. The acetylcholine always binds to nicotinic receptors, which are ligand-gated sodium channels. So this will cause the membrane potential to depolarize, you reach threshold, you generate an action potential. The action potential will then travel down here. So you'll notice the cell body of the postganglionic neuron is located within a ganglia, right here. And then the axons, which are relatively short, are going to travel to their various target structure. And here, the release of the neurotransmitter is always going to be acetylcholine. But the difference is the receptors found on these targets are going to be muscarinic receptors, which are G protein coupled receptors. And there are multiple subtypes of muscarinic receptors, like when we looked at the adrenergic receptors. That's why you can have various effects on targets. They could be excitatory or inhibitory depending on what particular muscarinic receptor is involved. So if you look here, I do want to distinguish the nicotinic from the muscarinic receptors. So nicotinic receptors are ion channels. That's what they are. So they're inotropic receptors. So this here is trying to represent acetylcholine. When that binds its receptor, this channel opens, and what's going to happen is sodium is going to want to go into the cell because of the concentration gradient. So I'm just going to want to enter. What we're going to get is depolarization. If you depolarize to threshold, you will have an action potential, and it would be excitatory. So the nicotinic receptors are always excitatory. So places that you'll find nicotinic receptors are always at that pre- and postganglionic neuron synapse. So here's your preganglionic neuron, here's my ganglion, postganglionic neuron. So this is a location where we're going to have the nicotinic receptors. When we talk about skeletal muscles at the neuromuscular junction, that also has nicotinic receptors. And also if this here was the adrenal medulla, that also would be involved is a nicotinic receptor. Now the muscarinic receptors, actually, and also I failed to mention, no, I didn't fail to mention, that's right, just ignore my mistake there. Now muscarinic receptors are going to be G protein coupled receptors. So those are examples of metabotropic receptors. When the neurotransmitter binds to its receptor, it will, via changes in metabolism with the cell, indirectly affect ion channels found in the membrane. Now they could be inhibitory effects or stimulatory effects. So here again, this represents acetylcholine. This one is acetylcholine. So on this, the example on the left, what this did was this happened to, say, open up potassium channels. And potassium wants to leave the cell because you have a high concentration of that ion inside the cell. So it wants to leave, which would hyperpolarize, getting further away from threshold and that would inhibit. So example of this would be an M2 muscarinic receptor. So we can get something like in the heart that would be like a slower heart rate. In over here, this one is showing you an excitatory effect. Is it could close up potassium channels, or it can open up, say, something like a sodium or calcium channels, which if they come in the cell, this is going to result in depolarization. Either of these could result in depolarization. So there's multiple subtypes of muscarinic receptors. And examples of where we'd find this type that would be excitatory would be smooth muscle, would be an example. Now there are multiple subtypes of muscarinic receptors. There's M1, M2, M3, M4. So I had previously mentioned in when we looked at the sympathetic nervous system that there were cholinergic fibers that innervated sweat glands, and I had mentioned the specific type of receptor that was involved because acetylcholine is released and it binds to a muscarinic receptor on the sweat glands. These ones were M1. Now M1, they will act via increasing calcium levels in the cell, via changes in IP3 and DHA. M1 and M3 are similar. The M2 and M4 types of muscarinic receptors, they tend to be inhibitory and they decrease cyclic AMP. So we can have muscarinic receptors like you notice here with the sympathetic nervous system, that one location, the sweat glands, but all of our postganglionic targets will have a muscarinic receptor, but depends on which specific one. Do we have an M1, M2, M3, M4? And as I go along periodically, I'll write it down which one so you can see how we can have various effects. Some places it could be excitatory, some places it could be inhibitory. So look at the various structures that are innervated by the parasympathetic nervous system. So the cranial division are going to innervate structures in the head and in the thoracic cavity and in the abdominal cavity because of the vagus. The sacral division is going to be innervated structures in the pelvic cavity as well as the lower abdominal cavity. So we're going to start first with those associated with the cranial division. Now the eyes, the iris of the eye, remember, has two muscles associated with it. Well, the parasympathetic fibers innervate the innermost layer, which is the pupillary constrictor muscle, which is also referred to as the pupillary sphincter muscle. When it contracts, it causes the pupils, or say the pupil diameter decreases, allows less light in the eye. So pupillary sphincter muscle contraction decreases the diameter of the pupil. Now this muscle is innervated by the oculomotor nerve. Now the ganglion involved for this target is called the ciliary ganglion. Now you remember ciliary ganglion, you're like ciliary. I remember ciliary having to do with the ciliary muscle of the eye, but that was associated with the lens, but that's going to be another structure we're going to look at. But the ciliary ganglion is location of postganglionic neurons that innervate the pupillary constrictor muscle. That ganglion also is going to have the fibers that innervate the ciliary muscles of the eye, and the ciliary muscles of the eye, in response to this, are going to contract. And what's going to happen when that contracts is the lens gets fatter, which is important when you're reading. So we're in rest or digest mode. So you need to have the lens to get fatter to bend those light rays back towards that fovea centralis, which has the location of the highest visual acuity. So both the pupillary sphincter muscle as well as the ciliary muscles have M3 muscarinic receptors, and those are going to be receptors going to be excitatory, so because both muscles will contract. both innervated by fibers from the oculomotor nerve. The salivary glands are also innervated by parasympathetic fibers. You think about it, rest and digest, you're going to eat. You need saliva being produced when you eat. Now you have two parotid glands, one on either side, close to your ear. Plus we also have what we call sublingual glands and submandibular glands. The submandibular and sublingual glands are going to be innervated by fibers from the facial nerve. And the facial nerve, see if I have this separate. So here's showing you the submandibular ganglion. And its fibers are going to be innervating the sublingual as well as the submandibular gland. And that's coming from your facial nerve. So as a response, you have an increase in salivation. Now the other salivary gland is the parotid gland, but that's innervated by a different cranial nerve. Oop, I don't have my picture. So here, the parotid salivary gland is innervated by the glossopharyngeal nerve. And the location of that ganglion is called the otic ganglion because it's located near, notice that here, it's located near your ear. So same thing with this one is you have an increase in salivation. Now the cranial nerve 7 also innervates other structures. We have something called the pterygopalatine ganglion. Well, this is a location of cell bodies of postganglionic neurons that are going to innervate the lacrimal glands, which you see here. That would increase tear production. It also innervates glands in the nasal cavity, so it increases nasal gland secretions. So with the, so you can add that even though it says lacrimal glands. But cranial nerve 7, that will, via the pterygopalatine ganglion, we're going to increase tear production as well as nasal gland secretions. Again, these are all the salivary glands as well as the lacrimal glands. They all have M3, muscarinic receptors, again because they're stimulatory. So we're increasing secretion or glandular secretion, and then some of them they were talking about contraction of muscles. Now the structures innervated by the vagal nerves, because remember we always have paired cranial nerves. This is, you'll notice there's in your notes, a lot of structures are innervated by the vagus. So let's start with the lungs. The lungs is going, because it has smooth muscle again. Now the smooth muscle of the lungs in response to vagal stimulation would result in I'm going to break it up, constriction, because it causes the smooth muscle to contract. But it also increases secretions. And these are all via M3 receptors. You can see that most often it's going to be M3. So this, when you're in rest or digest mode, the airways tend to be more narrow. You have a little bit more secretion. Now, most of you are never really going to notice this, but some people do, especially if you're asthmatic. It tends to be noticeable more when you're eating. You find it a little bit harder to breathe, and you're always having to cough up a little bit of phlegm. So some of you may notice this. I didn't notice it when I was younger, but now I kind of notice it more, that when I eat, sometimes I have increased phlegm production, so I have to cough up a little bit more. I don't notice as much about not being able to breathe. But this is relatively minor because there is not a huge amount of innervation, luckily, by the parasympathetic nervous system. The heart is also innervated. And here this picture includes what we showed you with the sympathetic nervous system, but the vagus does innervate the heart. And you'll notice it's primarily it's not the muscle, it's the components of what we call the cardiac conduction system. So the pacemaker, SA node. So the big thing it does is it decreases heart rate. So when you're resting, digesting, you'll notice that your heart rate is much lower than when you're running or stressed. So it does result in decreased heart rate. This one is via M2 muscarinic receptor. Now the digestive system, well innervated by the vagus, because we're in rest or digest mode. So you'll see a lot of things that are associated with the digestive system. So we have the gallbladder. Now the gallbladder stores bile, which is needed to aid in fat digestion. So what that will do is the gallbladder has smooth muscle associated with it. So it increases contraction of the gallbladder. But also what it does is, I'm going to look right in here, that hepatopancreatic sphincter, the sphincter of Oddi, which controls the exit of the bile as well as pancreatic enzymes going into the first part of the small intestine, is controlled by that. So you think about it, rest and digest mode, I want the bile, I need it to get into this structure here, so it will relax that sphincter of Oddi. Okay? From now on, I'm not going to include any receptors because it gets too much for you, but I just want to see that we can have different types of receptors. So we have different type of receptors in that sphincter of Oddi versus the gallbladder, because one's relaxing and the one's contracting. Now pancreas makes a lot of enzymes involved in digestion. So its effects on the pancreas is to increase secretions. So you increase all these enzymes being released. Again, you have to relax that sphincter of Oddi for those secretions to enter the small intestine. Then if we look also, and of course we're going backwards now, we should have got started with this, is the stomach. The stomach, in the stomach it will increase motility, movements in the stomach, increase secretions. So you see a common theme, we're increasing secretions. And what also it does is it relaxes. This will be a common thing that we see with the parasympathetic nervous system. It relaxes sphincters. So this sphincter right here that controls the exit of the what we refer to as chyme in the stomach, which is like an oatmeal-like paste after you're done mixing up the contents of what you ate. It controls the exit of the chyme into the small intestine. What it does, it will relax it, because we want things kind of moving along as we go through the digestive system. The small and large intestines are also going to be innervated by the vagus, but the sacral nerves actually are going to innervate those pelvic splanchnic nerves are going to innervate the latter parts of the large intestine. But the small intestine, as well as the first half of the large intestine, are innervated by the vagus. So in the small intestine, so pretty much I can just kind of circle similar things. So in the intestines, it increases secretions, it increases motility, and it relaxes sphincters. And one of the sphincters I want to mention is down here, the internal anal sphincter. The external anal sphincter is skeletal muscle, so that's innervated by somatic motor fibers. The internal sphincter is smooth muscle, and in regards to innervation from sacral nerves, because this will be innervated by sacral nerve because it's way too far down for it to be the vagus, is that will cause that to relax, which will be important in defecation. Other structure, which I just did that innervated by the sacral nerves, or those pelvic splanchnic nerves, which I see right here, pelvic nerves, is the urinary bladder. This, again, rest and digest, that's probably when you want to go and use the restroom. So here it increases contractions of the bladder, but it also, right in here, is it relaxes the internal urethral sphincter. So you notice with the, we have an internal and external urethral sphincter, and we have an internal and external anal sphincter. Both internal sphincters are smooth muscle, which would be controlled by the autonomic nervous system. The external sphincters are skeletal muscle. they have somatic innervation. They're coming from the pudendal nerve. So here, urinary bladder contractions, relax that internal urethral sphincter. But I want you to kind of think about it, why we have different innervation here is when your bladder fills up, thing is you don't want to automatically let that urine fall out because you want to be in an appropriate place. It's going to be important that we have voluntary control of these external sphincters, because that's extremely important. So there we have the somatic innervation there. The last thing we want to look at is the genitals. So in previous lecture about the sympathetic nervous system, we said a lot of the different components of the reproductive tract were innervated by sympathetic fibers. They were involved in emission, ejaculation, orgasm. But the parasympathetic fibers, which are coming from the sacral nerves, is that, you'll notice on this in the male, we follow the fibers, look at its innervating structure in the penis. Well, what it does, the corpora cavernosa is an erectile tissue in the male. When that becomes engorged with blood, you will have an erection. So the parasympathetic fibers are going to be involved in resulting in an erection, and this will also apply to the female because females have erectile tissue in the clitoris. So with the parasympathetic, you have erections. Sympathetics, you have emission and ejaculation. Way to remember it, the student taught me this and it actually works, is the parasympathetic, with a P, is responsible for pointing. Think erection, becomes erect. The sympathetic is involved in shooting because ejaculation is derived from a word that means to shoot forth. That's how you can remember it. Parasympathetic involved with pointing, sympathetic, I spelled sympathetic wrong, with shooting. That's how you can remember it. That was going to be something we're going to be talking about in lecture, where you have dual innervation of both components of the autonomic nervous system, where they're kind of working together in a way. Start off with parasympathetic, then you move to sympathetic. So we'll talk about that at a later time.